Every time after science.
Oh, after science, do you go out? The three of you?
Because we have extra time before we go to the game.
Oh, yeah. That's good. So like Drew does that on Mondays. When he finishes piano, he has a little gap before carpool for gymnastics. So he goes to Jersey Mike's. No, Jimmy John's. He goes to Jimmy John's and gets us the same sub every Monday. That's kind of his thing.
If you want to play some music from my video game on my tablet.
Time when we're
Just very quietly because they're doing class.
And always drive in Annabelle's car and we scream from the unicorns.
Pink fluffy, hey, Lucy.
Yeah, I got her
Oh, never mind. I
We
can't do it.
scream that, and of course it ends whenever we go by a person.
You look like you've been in the sun. You got a little bit of a tan. Were you outside?
2 soccer games.
Uh-huh. See, I don't do that. I just burn. I'm impressed. Yeah, you look like sun kissed. Nice.
I want.
We do it too
my
loud that
last
we can't even hear when the
basketball
music
game
starts.
yesterday.
We were talking about favorite restaurants. Do you have a favorite restaurant?
Not really, I just go to almost every restaurant.
Do you all, yeah, you're like me, you like them all. Yeah, yeah. What is yours? Chick-fil-A? Oh, that's easy. Chick-fil-A is easy. So there's this place. There's this place in Marietta, so I'm not at Marietta a lot, because Drew does gymnastics out there, and he hasn't had his license long enough to take his whole team, so we all still carpool because it's just easier. So, but I'm in Marietta like every Tuesday, right? And I found this authentic Mexican taco joint. I'm talking, it's in a shack in a parking lot. Like it looks like it maybe used to be like I don't know, like a photo development shack. It's a little tiny building, tiny building. You walk in, they have 2 tables in there. And the tables are this big, and they only have 2 chairs on each side. So it's little, it's a walk-up counter. But it is the best authentic Mexican tacos I have ever had. They give you the little styrofoam box, and it's like 2 of those small, like straight taco size corn tortillas, and they'll throw like the steak or the pork or whatever in there. And then they just throw like a handful of diced onions and like a handful of cilantro. Oh my goodness, it is so good, y'all. It's so good. It's my favorite.
I don't know what it's called, but we went there and you could watch the people like make meat.
Oh, like hibachi? Like they cook in front of you on the grill, or? Yes, I love hibachi.
I didn't like it because it was so hot.
Yeah, they put that flame in front of you, and it's right in front of your face. They make the little onion volcano. And put that onion volcano.
Baby, and we got so scared.
Oh no. I've seen babies at those places scream and cry because they're like, it's so loud and so bright all of a sudden. Those places are fun though. I like the hibachi places.
I went to a hibachi place one time with my friend Daddy and Rory.
I've never been to hibachi.
You gotta go. It's fun. That or like I was saying, Korean barbecue. The Korean barbecue place that's at Marketplace is really, really good.
I think someone my mom knew got food poisoning from a Korean barbecue.
Oh yeah, you gotta be careful with that. Yeah. You have to know what you're doing because they do give you raw meat and raw veggies. You gotta be careful. Yeah, you cook it yourself at the table.
Oh.
It's kind of fun. But like, so my anniversary was Sunday, was our anniversary. So we went to Bare Bones it's in Beaufort, downtown Beaufort. That's where Bare Bones is. And it was so freaking, everything they brought out was so freaking good. Like, I love a good ribeye, man. I love a ribeye. And it was like a big ribeye. I couldn't even finish it. I still have half of it in the fridge. I'm gonna have to eat it tomorrow night, I guess. It was delicious. Big old ribeye, man. It was good.
Yeah.
So we have how many more classes before our exam? 2, yeah, 2 in all today. And then next week, what are we doing? Next week is review, and then we have finals. Has everybody picked their elements for, oh yeah, I don't think you guys have.
Has anyone?
I don't think so. You can look at the thing and tell me. Go look at you'll have to look at the list and tell me. But I don't think anybody's picked hydrogen yet. But you can go look.
Because I was thinking like a superhero called Hydron.
Do it. Yeah. You've got like 3 weeks left really to get your final project together. Okay. Yeah, yours is done. It's done, because he's Mr. Get Ahead of the game. So yeah,
I'm just like the designer, Colin's just getting it more.
okay. So yeah, so you've got to get your final projects done. Be sure you get them ready and you're ready to present. Now, listen up, girls. I'm gonna put this in this week's email, but I'm reminding you, I'm gonna remind you now in person. When you look in the preface of your binder, like Lucy has it open right there. Lucy, turn like one more page. Let's see, maybe it's one more, I think. Can you turn one more? There's one thing, it says grading rubric. Maybe that was it. Yeah, that's it. You see that right there? Oh, no, one more after that. That's the info page. You've got it right there. One more page on the back, right there. See that? That's a grading rubric. So for those of you who don't know, that's how I'm gonna score your project, which means that you should be able to predict your grade before you even present. The only thing that's up in the air really is like how well you present, but none of you guys should have a problem presenting. So you should be able to predict what your grade will be. That tells you how to get 100. You do all the things that are on there and you follow all the steps, you'll get 100.
Well,
Yeah.
15 times 6 isn't 100.
There's an extra added, we get like 10 added in or whatever it is. Yeah, 10 points added in just for showing up. It's not on there, but it's on the actual grading sheet that I'm going to use for when I score it up. So anyway, are we ready to start our last lesson? This is our last lesson. Because next week's review. Everly looks excited. Are you excited? It's our last lesson, Everly. All right, Annabelle, are you ready? We'll put that up so we can be ready. All right, our very last lesson. What am I wearing? Yeah. All right, here we go. Let's see, what have we got? So today we are talking about nucleic acids, which are what make up DNA, right? Yeah, okay, yeah. So nucleic acids, what are they made of? Some kind of a chain, do we know? Do you know, Annabelle?
A's, T's, B's, and C's.
A's, T's, G's, and C's. Those are what we call our base nucleotides. So nucleic acids, you know?
We're made of phosphate, sugar, phosphate, sugar.
There you go. Phosphate, sugar chains. All right, look at this diagram right here. You see your G, C, T, A?
Yeah.
Those are what we call our nucleotide bases, right? Those are what pair on the opposite side of the helix. We talked about how DNA is like kind of sort of like a ladder that's twisted round and round on itself. It's very hard to model because it's a weird sort of a 3D, not a single helix, but a double helix. Like a ladder, if you just twisted it, would be a single helix, but it makes a double helix. And it's because of the way the molecules are shaped and how they bond. But see these pink things? What do you think the pink things represent?
Oxygen.
Well, those are pentagon shaped, so that tells you this is a carbon ring and there's an O in there. Those are the sugars. These are the sugars, see them?
Yeah.
And then what do you think the P's represent?
Those represent phosphorus. Phosphate.
Phosphate. So sugar, phosphate, sugar, phosphate, sugar, phosphate. You see them?
Yep.
So that right there is your nucleic acid. Sugar, phosphate, sugar, phosphate, sugar, phosphate. It's a chain like that. And then coming off of the sugars are those little nucleotide bases that pair up to make our genetic code, right? Everything about you, how you look, how your body functions, what proteins and enzymes you produce, all of that is in your DNA. There are only really two kinds of cells in your body that don't have your entire genetic code in them. Do you know what they might be?
They might.
So
Skin.
your skin has your entire genetic code. If I go like this, and somebody with a lab coat on that does forensics can come behind me, they can take that, swab it, take it to a lab and pull up my entire genetic code. It's that good now. It's called touch DNA. Just skin cells.
What about your hair?
Well, your hair isn't really alive. It's full of keratin and stuff. So it's not a living part of your body. That's why when you cut it, it doesn't hurt, right? There's no nerves or anything in it. So the two types of cells in your body that don't have your full genetic code are your red blood cells. They just don't.
Your what?
Your red blood cells. And then the other ones are your sex cells, your eggs or your sperm. They only have half your genetic code because they need to come together to make a full human being with a full genetic code. So they only have half. They're called haploid.
If it's
Ah, because it is rooted into your living skin, your living scalp that has all kinds of nerve endings in it, right? So it's a sugar phosphate chain with nitrogenous bases. Those are the GCA, nitrogenous nucleotide bases, connected to the sugar units. That's a good question. I would think it would be asparaguses, but asparagi maybe. I think it's just asparagus. Like moose is moose. You know what I mean? But you know what? That's weird. Like, think about it. When you eat corn for dinner, you don't eat a kernel of corn. You eat like a bunch of them, right? Or pea, but you say peas, but you don't say asparaguses. That's interesting. I have peas, or I have beets, or I have potatoes, but I have asparagus.
Maybe it's.
I'm gonna look this up now, Lucy. You've sent me on a wild goose chase.
Maybe it's asparagus.
Asparagus could be. All right. So DNA uses what kind of bases and forms a double helix? What kind? Four. Very good. It uses 4 bases and it forms a double helix, which is specific. What does A go with everybody?
T.
Everybody. Good. And then what does G go with? C, right? And you see how they made them like puzzle pieces that fit together? Because they really do fit that. I mean, not that exact shape, but they really do fit together physically. So that's why they bond. So if you look right here, these are components of nucleic acids. This is thymine. What do you suppose thymine is over here? T. T stands for thymine. And U stands for uracil, and that's the one that's in RNA, not DNA. Remember last week you had to change all those to use because that's uracil. So what do you suppose these 3 are right here? Right, guanine and cytosine. Yep, right. So these are your components of a nucleic acid. You have deoxyribose, phosphate, and ribose. These make up this part, this long part of the ladder. And then thymine, you're only going to find in DNA. Uracil, you're only going to find in RNA. And then adenine, guanine, and cytosine, you're going to find in both DNA and RNA. So replication of DNA only takes place when the helix splits. It basically unzips.
So is ribose only in RNA and deoxyribose?
Yes. Yes. DNA stands for deoxyribonucleic acid, and RNA stands for ribonucleic acid. Very good. Yep. So deoxyribose is only in DNA and ribose is only in RNA. That's exactly right. So we actually divide these A, T, G, and C into 2 different groups or categories. So we have pyrimidines and purines, and our pyrimidines are the T and the C, and our purines are the A and the G. And this just has to do with how they're shaped, how they pair, what they're composed of. And if you look, you can see that's how A and T bond. That's how G and C bond. And this is so cool, but you see this picture right here? I want you to imagine a zipper. You know how you have that piece that you grab and you pull and it opens up a zipper? In DNA, that is called helicase. And it goes through and it literally unzips the DNA. And as it unzips it, you have another strand that will come in and replicate as it goes along. And we'll talk about it a lot next year in biology, but that's how it works. It's really, really cool. So the arrangement of base what on DNA? What do we think? So this is triplets. Encodes genetic information by dictating the synthesis of proteins. Do you remember everything we did last week's lab was in sets of 3? And we call those triplets a what? Codon. Very good. The triplets are called a codon. And they all dictate which specific bases, those 3 bases together, are going to dictate a specific amino acid. And then that amino acid last week was your word, right? And then when you put the amino acids together in a certain order, it formed a sentence last week, and that would be your protein. It has to go in that order.
Stops and oc stops.
Yeah, so there are start and stop. Remember we had some last week that were start and stop. So your DNA will be reading, it'll be reading as it goes along, and it'll say start, and then it starts paying attention. And then it takes every 3 and produces amino acids. And when you produce amino acids in the right order, then you get a protein that makes sense.
So if you find out your DNA and then there's this kind of codons and then the codons will list out your name or something?
Not your name, but it will explain what which specific proteins it's producing. For example, like I have blue eyes, you have brown eyes. That's because there is in my genetic code and in your genetic code, it will say make this protein with more pigment. So in my case, it has almost no pigment because my eyes are very, very light blue and your eyes are a darker brown. So yours have more pigment. And it's just that it's telling it make more pigment because that's just the way yours is coded. Now, interestingly, there can be mistakes. Okay, and we'll talk about these next year, but we call them mutations. Isn't that a fun word? But you can have mutations where like one of your G's gets swapped out for a C or something happens and it's a little wrong. And sometimes those little things don't cause any problems at all. It's just one little letter wrong and your DNA is like, oh, I can make sense of the sentence without the one letter. You know, I understand. I understand what you're saying. And then there are others where it can cause someone to not be able to live. It can cause diseases. It can cause all kinds of problems. And it all depends on where the mistake is and what the mistake controls, whether it controls something that's absolutely essential or something that is maybe has multiple other things that are working with it so that it overcomes the mistake. But we use when we do we're going to talk about it a little bit today, but when we talk about like when they do crime scene DNA and things like that, they're looking for specific code, but sometimes what they're looking for are called SNPs or SNPs. And it's a single nucleotide polymorphism, which means you've got one or two little mistakes in your code. And those are specific to you because everybody has this gene and this gene reads like this. But in your case, these two letters are reversed and they can find you because you're the only person with those two letters reversed. Or it's inherited through your family that everybody in your family and your dad's family, whatever, has those reversed. So they look for all kinds of specific things. And sometimes those SNPs, as they call them, no problem. And you would never know. We all carry them. We all have tons of mutations and you would never know. And in some cases, it's very obvious because that SNP has caused a problem. Is it?
I should have watched that story box episode.
So if you look right here, it shows you like, here's your triplets, boom, boom, boom. So this is how DNA replicates. If it will go, come on, load it up.
Does that mean I'm
This is a graphic.
Does that mean I'm a mutant?
Oh, we're all mutants.
Well, there's real DNA.
It's kind of a little weird.
Living tissue.
Wait.
In most human cells, there is a structure called the nucleus. The nucleus contains the genome. In humans, the genome is split between 23 pairs of chromosomes. Each chromosome contains a long strand of DNA, tightly packaged around proteins called histones.
Histone.
Within the DNA are sections called genes. These genes contain the instructions for making proteins. When a gene is switched on, an enzyme called RNA polymerase attaches to the start of the gene. It moves along the DNA, making a strand of messenger RNA out of 3 bases in the nucleus. The DNA code determines the order in which the 3 bases are added to the messenger RNA. This process is called transcription. Before the messenger RNA can be used as a template for the production of proteins, it needs to be processed. This involves removing and adding sections of RNA. The messenger RNA then moves out of the nucleus into the cytoplasm. Protein factories in the cytoplasm, called ribosomes, bind to the messenger RNA. The ribosome reads the code in the messenger RNA to produce a chain made up of amino acids. There are 20 different types of amino acid. Transfer RNA molecules carry the amino acids to the ribosome. The messenger RNA is read 3 bases at a time. As each triplet is read, a transfer RNA delivers the corresponding amino acid. This is added to a growing chain of amino acids. Once the last amino acid has been added, the chain folds into a complex 3D shape to form the protein.
Isn't that wild? That's really complicated, that's happening inside your cells rapidly right now as you sit. It is just going on and going on.
Here is a cell, the basic unit of all living tissue.
What in the world?
In most human cells,
Hold on.
there is a structure.
Stat.
My mom just
Shoot.
learned about this. in her
Oh, did she? Oh, hold on.
college level things.
Nah, she's gonna learn about it at a much more, oh, that's what happened. Hang on, my, sorry, 1 second. It looks like that crashed. PowerPoint crashed. 1 second. So yeah, that's happening in your cells rapidly right now as you sit here. And it's happening millions and billions of times. Yes, sir.
Will the protein turn into a ribosome?
No, the ribosome is where your proteins are produced. It's where that's happening. Oh, come on.
They look alike.
They do. Yeah, because it just kind of showed it like a little blob. But oh, come on. It's right here. Why don't I see it? That is so weird. I'm not nuts, right? It didn't show up. Hang on, let's try that again. Share. What is the problem here? Oh, it's like everything's being wonky. Hold on. Sorry, guys. Technical. Here it is. Technical problems. There we go. Now let's see. We were right. Whoop. There we go. We're on this slide now. Hello. That was fun. So yeah. So you've got that's happening inside the ribosomes in your body all the time. It's taking your mRNA and it's translating it and it's spitting out proteins constantly happening all of the time in your body. That's how you're growing. That's how you're continuing to live. That's how you're replacing damaged cells. So let's talk for a minute about, oh, let me ask you this. This is a fun question. Do you know what our closest genetic relative is? And what animal is our closest genetic relative?
Monkeys.
It's a specific type of primate. It's a bonobo chimpanzee. They're genetically closest to us. What percent? I think I know this by heart, but I might be wrong. What percent of our DNA do you suppose you share with a bonobo? How much of your DNA do you think is the same as a bonobo? 74? What do y'all think?
84.
84? Oh, I was off by things. Actually, 98.7.
98.
And here's a good question. What percent of your DNA do you suppose you share with an apple?
An apple?
Yeah, an apple.
63.
63%?
Yeah.
You're pretty close. About, whoops, hang on. Whoops, oops. It didn't want to play. 40. So 40% of your DNA is the same as an apple. That just shows you that all living things are very similar in structure, right? All right, so let's look at this. Gene technologies. What do we do now that we have this knowledge of genetics and genes and how they function? What can we do with it? Well, this is real fun. Let's look at this first thing right here. This is DNA fingerprinting, and we've been doing this since the late 80s, early 90s. We will do this next year in biology. We will do a lab like this where you're going to get DNA and you have to run it through a process called electrophoresis, which will break the genes out into little markers like this.
Oh.
All right, so let's look at this graphic. So this is our crime scene victim.
So it's
Okay, let's say somebody was, I don't know, assaulted outside of a bank, because that's what's going to happen in our lab next year. He gets assaulted outside of a bank, and so they scrape under his nails because he says, I scratched my assailant, okay? So they scrape under his nails, they bring out all these skin cells, they test them. They also get some cells from this guy's mouth so they can compare his DNA to what they find. So from the mouth cells, this is our victim, okay?
So it's crime
And
section 2.
these are our crime scenes, and then these are our suspects. So if this is our victim and this was found at the crime scene, whose DNA is this? Right
The
here.
victim.
Yes, do you see that it's the same? Do you see that the markers are in the same place? That's the victim's DNA. And who did it?
Suspect 1 or 2.
Suspect 1 or 2. Well, look at the crime scene. Here's your crime scene DNA. Whoop. Yeah, it came back. So who do you think did it? Suspect 1 or suspect 2? Suspect 2? Yep. You see how it matches right here with the crime scene? See? So that's one thing we do with DNA. Another thing we do, I'm going to go over here first, is recombinant DNA. So we can actually take a bacteria, we can isolate a certain gene that we're dealing with, say it's some sort of medical therapy and we want to do gene therapy. We can actually take that gene, we can implant it in a bacteria so that it reproduces it, and we can grow, for example, like new plants that are disease resistant. We can use this whole process. This is very complex, but it's really cool. And then there's cloning. So the first mammal that was cloned was a sheep called Dolly, and it was in 1996. And I remember it well because I was in college getting my biology degree when it happened. So we were all just like wanting to know everything about it, right? So basically what they did is they took a mammary cell or cell out of the sheep's udder. This one sheep over here that we call her the donor. And they took her little cell, it was just a normal cell. And then they took her DNA and they had another egg donor. So they have a mammary cell donor and an egg donor. They took the egg from one sheep, took all the DNA out of it, took DNA from the mammary cell of another sheep, put it in that egg, and then implanted that egg into the uterus of a different sheep, and that the surrogate, and she gave birth to this little sheep named Dolly, and Dolly was a genetic clone of this other sheep. She was not produced through reproduction, sexual reproduction, the way sheep typically reproduce. She was cloned. It was a big deal because cloning, there's all kinds of ethical things, right? Is it good? Is it bad? There's definitely some good science behind it and things it can be used for, but it's also a little scary. So this is Dolly. It was a big deal when it happened. It was a real big deal. Why is everything so slow?
Any type of eyebrow. This
is for your eyebrows.
Actually trying to find the product that would take.
Lady, you got good eyebrow. What is going on today? I don't understand why everything is. Go.
Dolly the sheep was a sensation when she first appeared before the world. A scientific breakthrough, fascinating, but also menacing. Dolly was cloned by taking a body cell from sheep A, fusing that cell's DNA with an egg cell with its nucleus removed from sheep B. This fused cell developed into an embryo, which was placed in the uterus of a surrogate mother, sheep C. The resulting lamb, in this case Dolly, is a clone of sheep A. These were Dolly's so-called siblings, identical sisters, all cloned from the same sheep. Professor Kevin Sinclair worked with them and knew Dolly.
It was a step change in our understanding of early development, which ultimately led several years later in 2006 to the discovery that you can actually induce somatic cells. These are sort of cells that belong to the body to become embryonic-like cells that has the potential to divide into other cell types.
This was Dolly's greatest legacy, advances in what we call stem cell research. The ultimate, but still elusive goal is to use cloning technology to create healthy tissues which can be transplanted to heal damaged cells and organs. But back in 1997, Dolly seemed to raise more frightening issues. If we can clone a sheep, many asked, why not a human being? Since then, scientists have cloned more than 20 species, including horses and household pets. But scientists have also recoiled from using these technologies on humans.
I think where we draw the line is if we're going to use genetic manipulations, we have to be extremely careful, and we have to look for severe disease effects to control for. Because with a lot of these things, it's not just the person you're doing a genetic manipulation on or potentially cloning, but it would affect all the generations ahead.
So Dolly's brave new world has not quite turned out as predicted. A sheep created like none before her, but she could not escape her own mortality. She died in 2003, now stuffed in a Scottish museum. Barbie Phillips, Al Jazeera, London.
So we mentioned stem cells. Does anybody know what a stem cell is? So a stem cell is a cell that has not differentiated yet. So what that means is when a cell, some cells like skin cells are just going to divide and become more skin cells. But some cells are stem cells, meaning that they are, when they are formed, when they are, they divide off of another stem cell, it has to decide, am I going to be bone, blood, skin, pancreas, spleen? It can become any type of, or a certain type of cell. Oh yeah, yeah. It can become a different type of cell. So what's going on here is, this is fun. So who's in there? I can't see. I think I have a random. No, I have Downy Woodpecker in there. I think I can't see all of them. Okay, my little bird, my little bluebird house right here. Their babies hatched. I could hear them last night. They're chirping very quietly. So my male bluebird, who's the one that's really, really blue, the female's blue, but she's not as blue. She's he's very protective. So he keeps chasing everybody away from the feeder because his babies are right in here. So it's become quite a thing.
His babies are out in the bird feeder.
His babies are in the little birdhouse that's right there, hanging on a hook on the side of the deck. So he's being very protective of them. So he keeps chasing away people who are minding their own business, just trying to eat their breakfast. It's become quite a battle. So anyway, stem cells can become other kinds of cells. And stem cell research is very cutting edge because say you have liver cancer and you need to have your liver removed. Okay, liver cancer actually took my aunt's life this year. It's very bad. We lost my aunt to liver cancer. Say they were able to take some stem cells from her body and control them to grow liver. And say they were able to grow her a liver from those cells in a lab, a whole liver, and then take out her liver, her diseased liver, and put in a new one. That's the female bluebird right there. On top of the feeder. That's the girl, the mama. So there are all kinds of things that stem cell research can do for us that we're very curious about. So we're trying to figure out how to do that safely, ethically. So a stem cell is a cell that is not yet differentiated. DNA within a stem cell will tell it what to become. It might become skin, bone, liver. Isn't that cool that you have cells in your body that your DNA still has to tell it what it'll become? You can find stem cells in your bone marrow. A lot of them are there. And there's also a lot of them present embryonic in a forming embryo in a fetus. There's a lot of stem cells. Stem cell research is fascinating and complex, but not without the potential for harm. How would you feel if someone took your DNA and cloned you? Right? Are there human clones, natural human clones? There are. So you ladies are twins. But you're what kind of twins?
Identical.
Are you identical or fraternal? You're fraternal twins, which means there were 2 eggs, 2 sperm, 2 different sets of DNA, right? But I have some really good friends who are identical twins, which means there was 1 fertilized egg that split and made 2. So they are basically natural clones of each other. So y'all don't fall into that category, but like my friends Anna and Lily, they did. They are identical twins. So identical twins are like naturally occurring clones. Nobody took Anna and then when she was 4 and then made Lily. Like Anna and Lily were always, they developed together. Just one was like
friends, identical twins. I couldn't even tell them apart.
exactly.
And there's some players in a league
Mm-hmm.
who are identical twins.
Mm-hmm.
No one could tell them apart.
There are all kinds of there are different types of identical twins, but Anna and Lily, my friends, they're called mirror image twins, which means it's really funny. Like naturally, one of them has hair that parts on the left, the other has hair that parts on the right. The other one is right-handed, the other is left-handed. They are literally like looking in a mirror. And it wasn't until they were about 8 or 9 that their mom finally helped me out because they look exactly like. Now that they're older, they look a little different, but when they were little, their mom was like, Lily wears her bow or her barrette when you're looking at her on your left. And I was like, Lily left. Now I got it. When they stopped wearing bows, it messed me up. But when they wear little bows when they were little girls, I could tell them apart. So stem cell research is fascinating and complex, but not without the potential for harm. In the US, here are the rules about stem cell research in the US. The federal government doesn't ban it, but it limits the funding for it. Basically, this is so it doesn't go willy-nilly, right? We limit the funding, we limit, how much money can go towards this research. That tends to limit the research a little bit. Now, that's both good and bad. It's good because it allows us to control for some things, but it's bad because it slows down what could be potentially really useful and helpful research. So there's a mix there. The National Institutes of Health prohibits federal funding for deriving stem cells from human embryos. So the National Institutes of Health will not allow the government cannot give funding for getting cells from human embryos. So there's another thing there, because you can take cells from human embryos and then use them to do stem cell research. But we want to limit that for sure, because what we don't want is a market for human embryos. That's not a good thing, right? And then, however, research that doesn't destroy embryos is permitted. So so if you can get it without destroying the human, the embryo, the human, then that's good, because destroying humans is generally considered not so good, right? Right. Yeah, so state laws vary widely, with some states supporting stem cell research and others banning it.
Georgia.
I don't know where Georgia is on this. It really depends. So scientists often use leftover embryos from fertility clinics for research. Now, here's where things get sticky. So if you go to a fertility clinic because you're having trouble having a baby without some medical help, they'll harvest some eggs, they'll fertilize the eggs, they'll make all these little human embryos, and they'll put them basically in cryogenic freezing. Just recently, there was a baby born. I think the mother was born in 1991, the actual mother, but the embryo was produced and frozen, basically suspended in cryo freezing, like suspended life, suspended everything, I think 1992. And then that embryo was donated and implanted in the mom, and the mom delivered the baby, but really the baby is like about the same age as the mom, just had development suspended for decades. Because when you put them in the correct cryogenic freezing situation, they can stay there indefinitely because they're not developing, it's suspended animation, right? So here's the thing. When they do this, they don't harvest one egg because it's very expensive and it can be kind of risky. So you harvest a bunch of eggs and then you fertilize a bunch of eggs and then you get a bunch of embryos, and some survive, some don't survive. And those that survive, some will get implanted so the mom can become pregnant and have the baby. And some, they will freeze cryogenically, suspend animation. And now you have all these human embryos that are sitting in a deep freeze situation. Some can be donated, like this baby I just told you about, and became we have a baby, right? This lady had a baby, but the baby's been frozen for decades. Wild, right? But those that don't get donated, what do you do with them? And this is where ethics come in. It's a human embryo. So if you take it out and implant it in the right situation, it's going to develop into a full human being. If you take it out of the freeze, it's going to die. So it becomes a whole thing. So now there's they're going to take them out of freeze. They're going to die anyway. So why don't we use them for research? So there's that. So there's it becomes very sticky ethically. It depends on how you think religiously, how you think ethically, what you think the moral situation is. It gets very, very sticky and very involved. But one thing that can't be denied is that while the procedure to get the data can be questionable ethically, the data is very helpful. So what do we sacrifice? What do we not sacrifice, right? What?
Imagine
Yeah.
walk to two identical twins
Mm-hmm.
and tell a secret to them about the other twin.
Uh-huh.
But it's the other twin.
I saw a video just the other day. Apparently there are these Australian twins that run a pelican rescue, a seabird rescue, but they just recently made the news because their mom was carjacked, and they do that thing that some identical twins do where they speak together at the same time. It's really weird. Did you see it? It's going around the news. You can look it up, like Australian identical twins speak together, Google it. It is the wildest thing. Like they're like, our mom went up the hill and he said, get out of the car, and he had a gun. And it's like, they're going on and on. It's like, what is happening? It's so weird. Have you done it? Yep. Yep. So cloning. The federal government doesn't prohibit cloning, but it does prohibit using federal funds for cloning. Okay, so if you have privately funded lab, you can clone, but the federal government doesn't fund it, because where does the federal government get its money? From what? Taxes, from you, from me. And what if you personally don't agree with cloning? Do you want your money to go to cloning? No, so that's why the government is like, well, we won't fund it, but if private people want to fund it, that's fine. So that's different. And then, hold on, I'm clicking. This applies to both therapeutic and reproductive cloning. Policy changes. So in 2009, Obama overturned Bush's stem cell policy, which limited federal funding for stem cell research. He kind of opened the gates and was like, let's do more research. And then there's, I love this one, the Dickey-Wicker Amendment. That's a fun name, right? The Dickey-Wicker Amendment prohibits the destruction of embryos to create human embryonic stem cell lines. That's what the ES stands for. So you can't destroy a human embryo to create this line, is what the Dickey-Wicker Amendment says. All right, let's talk about energy in living systems. So we've talked about this before, photosynthesis and respiration. What do you notice about those equations?
They're reverse. Right?
They're what?
They're reverse.
Yeah, they're opposites, right? They're exactly the reverse of one another.
Heterotrophs.
Yes, yes. So you've got, yep, your cellular respiration. This is what happens inside your body, inside your cells. And then at the top is your photosynthesis. And if you look, they are reversed. Do you agree? Does everybody agree that they're reversed? Like what are the reactants in photosynthesis?
In photosynthesis.
What are the reactants?
It's C6,
What is
carbon,
CO2?
oxygen,
Carbon dioxide and
water,
Water. water.
Water.
What are the products in cellular respiration?
sugar,
Carbon dioxide and water, right? You see it there?
Yeah.
So that's why when you exhale carbon dioxide that you didn't take in, because it happened, it's produced in cellular respiration. So green plants use solar energy, carbon dioxide, and water to synthesize glucose during photosynthesis. The reverse of photosynthesis is cellular respiration, in which glucose is broken down into carbon dioxide and water. Energy is harvested in the process by the production of about 38 ATP ions per glucose molecule. ATP is adenosine triphosphate. That's your little energy molecule. So basically, there's this whole process. We'll learn about it next year. But this is where your energy is produced within your cells and how it is produced. It looks like this. It's very involved. See this thing, Krebs cycle? That's where your ATP and electron transport. So through the release of energy during the breaking of its third phosphate bond, ATP fuels life's processes, motion, synthesis, and transport. Everything you do, that energy is ATP. And this is where it gets produced within the mitochondria of a cell. And it's this whole process here, and it's very involved. Cellular respiration. Let's real quick look at this one. This is going to take your cellular respiration that we just talked about and show you how it works.
Why wait? Start your MD this September.
Yes, we should all be doctors.
Within weeks.
Oh, they go back and change it. Like they delete one. Yeah, they make a new one.
Person, one of us is, and one of us is that. Mainly because when I wake up in the morning, it just takes a while for me to get my energy back. It takes a lot of time and coffee for that to happen for me. Girls really don't have that luxury. They are busy performing cell processes all the time. Active transport of many substances needed for their survival, for example. And the energy currency they need, specifically, is ATP. ATP stands for adenosine triphosphate. It's a type of nucleic acid, actually, and it is action-packed with 3 phosphates. We have a video all about ATP and how it works as an energy currency. So where am I going with this? Well, cells have to make this ATP. It doesn't really matter what kind of cell you are. Prokaryote or eukaryote, you have to make ATP. The process for making that ATP can be different. depending on that type of cell. One way is called aerobic cellular respiration. Lots of organisms can do aerobic cellular respiration. But this video is specifically going to go into aerobic cellular respiration in eukaryotic cells.
That would be you.
That means this video is talking about the process within cells that have membrane-bound organelles, such as the nucleus and mitochondria. Eukaryotic cells include the cells you find in protists, fungi, animals, and plants. The mitochondria, which can be found in most eukaryotic cells, are going to be kind of a big deal in this aerobic cellular respiration because some of the process occurs in the mitochondria. So let's get started. Remember, the major goal for any organism performing this is to make ATP. Okay, here's the overall look at the equation for aerobic cellular respiration. Remember that reactants, inputs, are on the left side of the arrow, and products, outputs, are on the right side of the arrow. This equation, by the way, is remarkably similar to photosynthesis. Look at how the reactants and products are on different sides. Well, that doesn't mean they're simply opposites. It does show that they have substances in common. In photosynthesis, organisms make glucose. Notice how glucose is. But in cellular respiration, organisms break the glucose down to make ATP. Fun fact, you know how when the bean seed cluster makes it grounded, it isn't easy to do photosynthesis yet?
What I'm gonna do, because I'm watching for our time, is I'm actually gonna pause this one. It's in your you don't need it till next year, and it's in your resources. I'm gonna zip through these last couple, okay? So let's talk viruses for a minute. I'm not gonna show you this one because we're low on time, but how a virus replicates is a virus has to enter a cell and basically hijack its it hijacks the way your cells make DNA to replicate its own DNA. Now here's a question. Is a virus alive?
Yep.
Nope. Viruses are not living things. They are like strands of DNA, but they aren't alive in the way a bacterial cell is alive. So if you get strep throat, anybody ever had strep?
Yeah.
Had strep before? That's a bacterial infection. Those are microorganisms.
Strep tests are one of my worst.
Oh, I hate it. Well, right? So strep is a bacteria. Streptococcus is a type of bacteria. And you can kill the bacteria by taking an antibiotic, right? Antibiotic, break that word down. Anti, not, right?
Like
Bio,
living.
living. Antibiotic is going to kill the living bacteria. One sec. But viruses aren't alive. When you take an antiviral, all it's really doing is inhibiting its ability to replicate. It doesn't kill it. Yes.
Me and my older brother got strep throat on vacation once.
That's not fun.
It really sucks.
It does.
We
So
don't get to go anywhere.
viruses
Just
use
stay
a
in the house.
host cell to replicate. We come in contact with viruses every day. The common cold is caused by a virus. So when you get a cold, it's just a virus. You can treat the symptoms, but it is not alive, so you can't take medication to kill it, like if you had a bacterial infection. Antiviral medication inhibit replication. That's what it does. It makes it harder for it to replicate. If you have strep, you can take an antibiotic and you'll soon be better because the antibiotic will kill the bacteria that's making you sick. If you get the flu, you can treat the symptoms of your while your immune system does its work, and you can take certain medications like Tamiflu to help prevent the virus from replicating, but you can't kill it because it's not alive. Some viruses are bothersome, some are deadly. HIV or human immunodeficiency virus develops into a syndrome called AIDS, which is acquired immunodeficiency syndrome that can kill you. Nowadays, not very often because we've got medications. So you can catch some viruses by inhaling someone's cough, but HIV, for example, you only get through body fluids. So that's sexual contact, sharing needles, mom to fetus, breast milk, or blood transfusion. And nowadays, I'm going to skip that one too. That's just showing you a particular new antiviral they're using for HIV AIDS. But very rarely now does AIDS get transmitted through breast milk, mom to fetus, or blood transfusion, almost never, because we test for it now before we do transfusions. But primarily it's sexual contact and sharing needles, drug use, sharing needles. Hold on, we'll come back. So we're not gonna have time to do a little research, so I'm just gonna tell you. But real quick, because we're wrapping up, we've only got 2 minutes, Lucy. So DNA phenotyping, this is cutting edge forensic science, cutting edge. This has only been around about 6 years. So what it is the main company that started this, now there's a couple that do it, but these guys are the flagship. It's called Parabon Nanolabs. And what they do is they can take DNA from a cell, say it's a cell found, or say it's DNA found at a crime scene and you're looking for the perpetrator, or say you found remains, somebody's remains, and you don't know who this dead body is. Like sometimes that happens. We have a lot of unidentified bodies people find, you don't know who it is. And it'll go on for a long time. So what they do then is they can take that DNA, and this company has done so much research and created such an efficient database that they can put your DNA, plug it into their system, and it will spit out a computer-generated picture of you, your face, based on your DNA. So for example, this was an unidentified body, and we didn't know who she was. They had to figure out who she was. This was the phenotype, the DNA phenotype that was created of her. That's the actual girl. Pretty close, right? Once they identified her, pretty close. And then look at this guy. This is an interesting one. This guy right here, that's the picture, but that's him in real life, because they try to put you, when they do the phenotyping, they put you at about 30 years old. They don't know how old you are based on your DNA. They just know what you look like. So they put everybody at the same age. So he was actually considerably older when they found him, but I have seen younger pictures of that guy and it was spot on. So Parabon says that since 2018, police have solved more than 200 cases with the help of genetic genealogy and phenotyping methods. Company declined to disclose the total number of cases, which they use because they're doing ongoing investigations. She was unidentified, and now she's been identified.
The picture that is a dead body?
No, this is her in real life. Those were living pictures of her that her family had when they finally discovered who she was. Because of that picture, they put it in, there's databases you can go to that have like DNA Doe Project. And they're like, we're looking for someone that looks like this. And her family or whoever was like, we're missing somebody who looks like this. So then they test the DNA and find out it's 2 families. Yep. Yeah, no, this is her phenotype. That's what they created of her. The computer did that. So now there's even more labs, including DNA International, Bodhi Technology, Othram, and the Center for Human Identification. That is in your resources if you want to watch it. It's really cool. And boom, you need this. Yes, ma'am.
The plural for asparagus?
You can do it in a minute. So take a minute to be sure that you see this. This is your Unit 9. Unit 9 test, right? Unit 9 test. Come on in, guys. Don't touch lab stuff yet. Let me talk to you first.
Yeah, I need to prepare a lot before I do
Asparagus. Could be. Could be asparagus. Asparagus.